Thank you, Hannah. It's my great pleasure today to introduce Dr. Erin Maloney. Erin is a postdoc that's working on a cooperative agreement between the University of Minnesota and the EPA. So we're so excited to have her here, and she's really helping build these bridges between the EPA and the University of Minnesota, which is so valuable for us. She did her BS at the University of Guelph and her PhD at the University of Saskatoon, both in Canada. And I would also add that she's here today. She's another speaker that we put to a lot of hard work. So she helped run an early career workshop with graduate students from IBS, WRS, chemistry, and earth and environmental sciences this afternoon with Lisa DeGuire, who I don't think could say, but also Nate Polish and Courtney Larson, who are here in the audience. And so you kind of embody the spirit of interacting and building community. So we're so excited to have you here. And I'm going to turn it over to you for your talk.
Okay, great. Thanks, Jennifer, for that lovely introduction. All right, just jumping the gun before your applause. All right, thanks everyone for joining me today. I'm going to be talking about some of the research we've been doing employing weight of evidence to identify priority chemicals across the Great Lakes estuaries. So in case none of that means anything to you, we're going to be going through what exactly that all entails, and you guys are all going to be experts by the end. So let's stick with it. So anthropogenic or human-related activities such as agriculture, industry, urbanization, and wastewater output have resulted in the introduction of numerous chemicals of varying chemical classes into freshwater systems. So for example, in the Great Lakes alone, between 2009 and 2018, over 550 unique chemicals were detected, including fuel byproducts, industrial chemicals, wastewater indicators, pesticides, pharmaceuticals, and personal care products. So these chemicals encompass both legacy pollutants. So these are chemicals that are released through historical operations that are no longer occurring, and also chemicals of emerging concern. So these are chemicals that are being continuously introduced into the environment that are still undergoing ecological risk assessments. So due to this continuous anthropogenic or human-related activity, alongside recent advancements in environmental monitoring technologies, there's increasing numbers of contaminants detected in the aquatic environment. This means that we have more chemicals to deal with than ever. However, environmental managers and research agencies are often faced with limited resources, which can make it difficult to determine out of all the chemicals that we detect, which we should really focus on and focus all of our kind of efforts and agencies on. Therefore, chemicals have to be prioritized for efficient implementation of research and regulatory actions. This means that of all the chemicals potentially detected in the water, sediment, or air, we need to determine a target group that we need to focus our research and regulatory actions on. So this brings about kind of prioritization. So there are a few different traditional prioritization strategies that have been employed in ecotoxicology. This isn't a lecture on prioritization, so we're not going to be going through all of them, but I'll point out a couple that are kind of been widely used over the last few years. So one of them is the PBT strategy, and this is where we look at persistence, bioaccumulation, and toxicity of the chemical we're looking at prioritizing. So persistence is a measure of how long a chemical remains in the environment, with chemicals remaining in the environment for longer being those of higher priority than chemicals that tend to degrade faster. Bioaccumulation means we look at how likely it is that a chemical will move up into aquatic organism inhabiting that system. And obviously, with more bioaccumulative substances, we care about those more than non-bioaccumulative substances. And then obviously toxicity is where we measure how likely it is that this chemical is going to cause adverse effects on the environment. So we look at the estimated or experimental properties, either estimated through programs or determined in the lab, and we determine whether they're persistent, bioaccumulative, or toxic, compare them to thresholds, and then we prioritize chemicals exceeding those thresholds. Another strategy focuses more on effects. So this is ecotoxicological thresholds of concern, or EcoTTC. And this is where we take toxicity data, whether it be measured in the lab or estimated or extrapolated, And then we derive this predicted no effect concentration. So a concentration that's unlikely to elicit an effect in an exposed organism. We then take groups of chemicals, group them together based off of structural similarity, mode of action, or functional use, and then derive this distribution you see up here, from which we can derive an estimated concentration at which there should be limited effect. That's the EcoTTC. Any chemicals with concentrations exceeding that would be priority chemicals within our data set. So these traditional prioritization strategies primarily utilize these classic metrics of environmental risk, including aquatic half-lives, bioaccumulation, and estimated or measured toxicity. And typically these are lab derived. And with toxicity data, a lot of the times we use full animals to derive those benchmarks. So here comes new approach methodologies, or NAMs. So these are kind of new techniques that are adding an extra dimension to ecological assessment. NAMs are traditionally known as these non-animal based approaches, which can be used to provide additional information for chemical hazard and risk assessment. So there are a variety of NAMs that exist, and I'm just highlighting a few here. They include kind of high throughput in vitro testing, so cell-based testing assays that would estimate kind of potential cumulative effect of a chemical. They're quantitative structure activity relationships, so programs that we use structure and physicochemical properties in order to estimate environmental fate or toxicity. There's ecotoxicological extrapolation tools, where we take the toxicity in one organism and we extrapolate it to another organism using similarity of the two organisms biologically. And there's also programs like adverse outcome pathways, where we try and link molecular-based effects to whole organism or ecosystem level effects, and look at our chemicals on a molecular level to try and estimate risk. So weight of evidence is a way to merge these new and traditional approach methodologies and use it them together for chemical prioritization. So weight of evidence is where we use a combination of information from several independent sources to get a sufficient evidence to give us to help us fulfill an information requirement, which is kind of vague, but mainly it means that we look at multiple lines of evidence, amalgamate that evidence together, and use that in order to figure out what chemicals we should be looking at in a priority context. So if we look at this kind of process in the formation of chemical prioritization, we take our problem, which is the amount of chemicals we have in the environment as our target groups. We collect all of our relevant lines of evidence. We establish knowledge gaps. We try and fill those knowledge gaps. We look at how reliable our data is and eliminate unreliable data. We weight all different lines of evidence, and then we amalgamate it to identify our final priority chemicals. Some advantages of weight of evidence for chemical prioritization is it improves our chemical coverage. So if we're looking at multiple lines of evidence, we can improve the coverage because maybe some chemicals have certain lines of evidence available and maybe others have other lines of evidence available. And we're able to kind of improve our scope. It integrates multiple different facets of risk, which might mitigate uncertainty associated with their ultimate results. And it kind of gives us a holistic estimation of environmental risk when we're looking at how a chemical interacts under different circumstances. So I'm going to be talking a lot about chemical prioritization, and I'm going to do this using two different case studies to illustrate one big point. The first case study, we're going to be looking at some data we used from the Milwaukee Estuary in Milwaukee, Wisconsin, collected between 2017 and 2018. And in the second case study, we're going to be looking at data we derived from across the Great Lakes estuaries from 2009 to 2018. The major differences in these two case studies is the first case study is obviously localized. We're just looking at Milwaukee, but we're estimating mixture toxicity and integrating multiple complex lines of evidence. So this is localized but complex. The second obviously is national and highly complex in its scope, but it's simplified in terms of its individual chemical prioritization. So we have localized and complex, national, gigantic, but a little bit simplified. And we'll show how we can use similar techniques to come out with the results in both of these. The ultimate aim is to show you guys how weight of evidence, new approach methodologies, and publicly accessible tools can be used to efficiently prioritize large chemical data sets and how those results can be used to provide recommendations for risk assessment and regulatory agencies. Okay, so let's start with our first case study. This is in the Milwaukee Estuary around Milwaukee, Wisconsin. This area was designated as an area of concern in 1987, mainly due to legacy pollution associated with industrial operations in the region. And this area of concern covers a confluence of Menominee, Milwaukee, and Kinnickinnic rivers and Lake Michigan, mainly around the major urban area of Milwaukee. The legacy contamination or legacy contaminants mostly come from industrial operations and include PCBs or polychlorinated biphenyls and polyaromatic hydrocarbons, as well as heavy metals. And the contaminants of emerging concern in the region mainly arise from intense urbanization, industrial activity, and agricultural activity slightly outside of the urban area. So this means that we have complex mixtures that may be eliciting adverse responses within the Milwaukee Estuary region. Our Milwaukee Estuary prioritization effort encompasses both individual chemicals and mixtures. So overall, we've done all this work, but I'm going to be focusing just on our mixture assessment. So if you break this down, you can see that we look at multiple different lines of evidence in order to determine which mixtures are of potential concern within the watershed. Our mixture assessment includes an evaluation of whole mixture effects. So what is actually happening with whole mixtures in the Milwaukee Estuary? It involves breaking down the chemicals into individual chemicals and chemical groups and assessing them for toxicological potential by evaluating in vivo effects, in vitro effects, and pharmaceutical potential. And it involves carrying out these predictive analyses where we try and merge the whole mixture effects we saw in Milwaukee with the toxicological potential we estimated to determine whether there are important drivers of effect that we're not seeing under those other two lines of evidence. So we'll start with how we evaluated whole mixture toxicity. We did this using caged fish studies across the Milwaukee Estuary. So these were carried out between 2017 and 2018 at 8 to 11 sites. And what we ended up doing was deploying adult fathead minnows at sites for 96 hours across the Milwaukee Estuary. So these adult fathead minnows, oh, I do have a pointer. Okay, this is great. All right. So these fathead minnows were deployed in these cage systems as pictured here, whereby a cement block keeps the cage system anchored to the sediment in a specific site. The buoy keeps the cages floating about halfway up the water column. And the cages contain 12 fathead minnows each, 6 males and 6 females, for a total of 24 fish per site. This is a picture of the cages in the actual water system. At each site, there's also an auto sampler, which automatically collects water samples over the course of the 96-hour period, which we can use for further analysis. So after the 96-hour exposure, we collect these fish, we collect serum, liver, and intestinal tissue, and evaluate in vivo effects or effects within the fish organisms. We also collect our water samples and use that for chemical analysis and in vitro or cell-based assessments. So our in vivo assessments included gene expression in liver and intestines using qPCR, and steroid hormone quantification using radioimmunoassays in the serum. Our chemical analysis included quantification of nutrient, wastewater indicator, and pharmaceutical concentrations. And our in vitro analysis involved evaluating bioactivity through a series of adagene assays and evaluating estrogenicity through T47DK Gluc assays. So let's get into our whole mixture results. What did we see? Well, over the course of the 2 study years, we found 80 unique compounds detected in the Milwaukee Estuary. In 2017, we found 41 compounds in at least one site. And in 2018, we found 76 compounds in at least one site. And looking at this chemistry data, we were able to group our sites into 2 general groups per year. The first were the sites that were more involved with industrial compound and wastewater input exposure. So we had distinct groups of chemicals with industrial compounds like polycyclic aromatic hydrocarbons. And our second group of sites were those with higher concentrations of pharmaceuticals and personal care products. So these 2 groups of sites will make sense when we look further at the in vivo and in vitro effects measured on a whole mixture basis. So when we're looking at these in vitro and in vivo effects, I'm showing you this very beautiful heat map here, which essentially shows you scaled effects across all of our measured cell-based assays, with purple representing not measured effects and yellow representing the highest measured effect possible. I've also broken these down into xenobiotic metabolism related and endocrine related effects, so you can kind of see trends across sites. Now, when we're looking at in vitro activity, we found that there was elevated xenobiotic metabolism bioactivity at all of our industrial compound dominated sites. So we're seeing that this trend with xenobiotic metabolism activity and industrial compounds kind of holds across this in vitro bioassay evaluation. However, we didn't see any site-specific trends in endocrine-related activity, indicating to us that there's no site-specific effects occurring there. When we looked at our whole mixture in vivo effects, so effects measured in the actual fish species, we found that we also saw some trends that were kind of a little bit more nuanced than our previous trends. So we saw elevated hepatic xenobiotic metabolism activity at industrial compound-related sites and elevated xenobiotic metabolism-related activity in the intestines at the sites that had higher concentrations of pharmaceuticals and personal care products. So remember, these are just trends. And we're kind of incorporating this into the weight of evidence evaluation of the region. We saw limited endocrine-related effects across the watershed and no effects on body condition. So that's what we saw with the whole mixtures. What happens when we break down the chemicals into individual compounds and mixtures and evaluate them on a toxicological potential basis? So we evaluate toxicological potential using this technique known as maximum cumulative ratios. And this is just a way to identify mixtures or chemical mixture groups that are contributing to the highest proportion of toxicity within a complex mixture. So it's essentially a ratio of either chemical or group to overall mixture. And you identify those important constituents by identifying those contributing the most to the mixture toxicity. So we do this by calculating toxicity quotients of individual chemicals, meaning we divide a chemical concentration by a benchmark of interest. And then we derive toxicity quotients of the mixture, meaning we add up all the toxicity quotients of a chemical at a site, and then derive an NCR for an individual chemical by dividing the toxicity quotient of the individual chemical by the mixture. Simple enough, right? So we take our chemicals and our groups contributing to greater than 10% of the mixture effect as important drivers within that individual site. And then we evaluate all of the important effect drivers across all sites to see if anything interesting is going on within our data set. So the first line of evidence that we use this technique on is using in vivo ecotoxicological potential. And for each of these slides, when I'm talking about the individual lines of evidence, I'm going to point out the databases that we use, which are publicly accessible, and the data, which include the benchmark on that last slide, and the grouping. So the way that we define chemicals into groups and assess them on a mixture group basis. So for in vivo ecotoxicological potential, we use the Ecotox knowledge base and the Compact dashboard. The Ecotox knowledge base is an open access database with a bunch of in vivo toxicity data collected from different studies. And the Compact dashboard is a series of publicly available databases, including applications that have data about chemical structure, individual characteristics, and toxicity. From these two databases, we derive 96 median lethal concentrations for fathead minnows. So the concentration estimated to kill about 50% of the population. And we use that as our benchmark for our MCR analysis. We grouped our chemicals based off of mechanism of action, structural and physical characteristics. So how similar they were in terms of target, structure, and activity in the environment. The next line of evidence we evaluated was our in vitro ecotoxicological potential. So we used data from an in vitro database known as ToxCast. And this is just a repository of all this high throughput screening data describing in vitro effects of chemicals on cells, protein, DNA, RNA, mitochondria, receptors, and enzymes. For this, we used our minimum activity concentrations at cutoff derived for chemical assay combinations as our benchmark. So what does that mean? So for an individual high throughput assay that we were given for a specific chemical in an assay, we identify the concentration at which kind of the activity starts, and then we use that as our benchmark for a cutoff for in vitro activity. We also grouped our chemicals based off of their molecular target and assay direction. So whether they're likely to cause a significant activity in molecular target and what direction that activity is, so activation or inhibition. Finally, we evaluated pharmaceutical potential using a series of different databases. So this is the most kind of data intensive one. We use this MapFast database, which includes some pharmacological data about all of our available pharmaceuticals. We use DrugBig, which is a database where you get to determine the target and direction of action of a specific pharmaceutical. And we use this David database, which is a way to group your pharmaceuticals based off of mode of action with pharmaceutical targets based on mode of action and gene similarity within an organism of interest. And in this case, David is using mammalian data. Okay, so our data for our cutoff was a peak serum pharmaceutical concentrations following dosing. So this is a common pharmacological metric used to compare potency between two pharmaceuticals. So it essentially means the highest concentration that a drug will reach in your bloodstream before it starts to get eliminated. These chemicals were grouped based off of their molecular target of the pharmaceutical and the direction of pharmacological action. All right, so those are our 3 lines of evidence. Those are our 3 kind of benchmarks, data benchmarks, and this is what we found within that assessment. So if you look at the Ecotoxicological Potential assessment, you can see across sites, we have some pretty common trends in what is driving this toxicological potential. So on an individual compound basis, which is always on the left, you can see that we have 2 main drivers of ecotoxicological potential. This triphenyl phosphate, which is a organophosphate flame retardant, and fluoranthene, which is a polycyclic aromatic hydrocarbon. So across all sites, pretty clear trend there. If we look at mixture groups, so when we're grouping our chemicals, you can see that mostly triphenyl phosphate, which was in a group on its own, and these polycyclic aromatic hydrocarbons as a general group were driving toxicological potential across these sites in Milwaukee. When we looked at a different line of evidence, however, we got different drivers of potential toxicity using our cell-based results. So if we look at in vitro activity, it indicates to us that the most prevalent drivers of toxicity on an individual basis could be bisphenol A, a plasticizer, metolachlor, an herbicide, or another organophosphate flame retardant, tris-2-butoxyethyl phosphate. And if you look at groups, it indicates that most of the activity is dominated by estrogen-related and Bregnane X receptor-related groups. So different results based off of what benchmark we're looking at here. If you want to get more confused, we'll move on to the pharmaceutical potential because we have, again, different drivers of pharmaceutical potential across all the data sets. So again, we're identifying a couple different drivers, including 2 individual compound drivers, desvenlafaxine, which is a antidepressant, and nicotine. And across the groups, we're looking at sodium ion channel activators and neuroactive agonists. So we then got that kind of confusing multi-line of evidence idea of what might be potential drivers within the Milwaukee Estuary. And we're looking for a tiebreaker. And this tiebreaker hopefully will come in the form of predictive analysis. So in predictive analysis, we use random forest regression and conditional permutation importance to link effects, which are in vivo and in vitro effects measured in the cage fish studies, and individual predictors, which are chemicals and chemical groups identified in our toxicological, well, just across the Milwaukee Estuary. And so basically what we do is we put all this data in, it kind of matches different combinations, and we get the set of predictors and effects with the lowest error rate, and that is ultimately our set of predictors and effects that we consider. However, we ran these random forest models individually for each of our in vivo and in vitro effects, so that we get a set of predictors for which we can use weight of evidence to identify if there's any kind of credence in the importance of these driving effects across the Milwaukee Estuary. To keep it a little bit simpler, we divided these effects into endocrine-related and xenobiotic metabolism-related to give some kind of weight to our weight of evidence. So this is kind of what it looks like when we pretty it all up and take away the numbers. The yellow are the important predictors. The black means that they weren't important. And there's also probably 150 other predictors that aren't included that were never selected as important in this analysis. As you can see, when we break it down into xenobiotic metabolism related and endocrine related, there are some compounds that pop up more than once for individual effects. So here are the compounds that popped up at least more than once across xenobiotic metabolism related and endocrine related effects. So we're seeing a couple single compounds, acetaminophen, anthraquinone, metformin. We're seeing some gabapentin. We're also seeing a bunch of different groups. And some of them interact with some of the stuff we saw before and some of them don't. So what does this mean when we kind of put it together and try to identify targets for further evaluation? Well, if we mesh together all of the output from the random forest regression, and we mesh together all of the toxicological potential output, and we mesh together some of our measured in vivo and in vitro effects, we can identify at least a smaller list of potentially important chemicals or mixtures within the Milwaukee estuary. And that was our goal, right? Get it smaller. So we identified some pharmaceuticals and personal care products that might be individual effect drivers, some wastewater indicators, one pesticide or metolachlor, one industrial compound fluoranthene, and about 7 mixture groups, which should be of further interest for evaluation. When we break these groups down a little bit more, we tend to see that there's kind of 5 classifications that tend to pop up, including polycyclic aromatic hydrocarbons, organophosphate flame retardants, pesticides, and pharmaceutical and personal care products. So this is a bit of a list, right? But we also have to point out that there were 113 mixtures and 37 individual compounds that never popped up in any of our analyses. And so we can eliminate those. So yes, we have weighted down our list to a set of targets for which we have to do a bit more research in terms of figuring it out. But we have effectively prioritized this Milwaukee estuary data based off of mixture effects. So has everyone got that? All right. Then we're going to move on to our second case study, which should be a little bit more straightforward, hopefully, but we'll see how I explain it. Okay, so this is the data across the Great Lakes estuaries. And this is a multi-agency environmental monitoring effort to characterize chemical concentrations across the US Great Lakes. So it doesn't include the Canadian side, it's only the US side, but it's a whole lot of data. It covers 9 years, 303 unique sites across 5 Great Lakes and associated estuaries. At each site, there was between 62 and 557 chemicals monitored. And there were 2 different sampling types, which is important when we kind of think forward in terms of this rest of this presentation. There was grab or composite and passive samplers. So the grab or composite are kind of short-term samples that just capture a very short period of time. And the passive samplers are these samplers that we put in the water and they allow to collect chemicals through passive diffusion. So we do get a different data set of chemicals for these 2 different sampling types. So you'll see the difference as we go through, but just note that these 2 are different. So I've said this before, but this included 550 unique chemicals. So this is what we're dealing with in this data set. So this would look familiar. This is our weight of evidence evaluation, this big spider web of different lines of evidence that we consider when we're trying to figure out what are the targets for further research and regulatory actions. We'll start with 3 of these individual lines of evidence: toxicological potential, which you can tell in this case is much more complicated than we looked at before, environmental fate, and detection characteristics. So for toxicological potential, we evaluated our chemical against 12 benchmarks, kind of wrapped up into several different groups. So some of these groups will look pretty familiar as we talked about them in our Milwaukee estuary effort. The first of which is not familiar, this is new. So these are water quality benchmarks, which we broadly define as either water quality criteria or screening values. The water quality criteria are current US, Canadian, and European federal and state or provincial guidelines. And screening values are either produced by non-governmental organizations or our previous guidelines, or are produced by non-water quality bodies of government. So they're non-official, they're just estimates of potential effect. The next group of benchmarks are benchmarks estimating in vivo effect concentrations. So these will look familiar. We derived benchmarks from the Ecotox knowledge base, looking at lethal concentrations across organisms and non-lethal concentrations across organisms. We broadly define these as apical and non-apical effect concentrations, with apical effects being for reproduction, mortality, behavior, anything that would be like really, really bad if it happened to a fish, and non-apical being something that would may be bad, but is not directly linked to the fish dying or the fish population decreasing. We also used QSARs, so those kind of mathematical equations that we talked about before, to derive estimates of toxicity to try and fill our data gaps. We also looked at pharmaceutical potential. So again, those maximum serum concentrations from the MATFAST database. and in vitro potential by looking at the in vitro activity concentrations from ToxCast, which we talked about, and cytotoxic burst concentrations from the CompTox dashboard, which we didn't talk about, but they're essentially concentrations likely to cause cytotoxicity in the cells. So these are our different benchmarks. And for each of these individual benchmarks, we evaluated them by computing a toxicity quotient and comparing it to detection frequency. So again, just to remind you, we take our toxicity quotient by dividing our chemical concentration, which in this case was the maximum concentration across the Great Lakes, against a benchmark concentration, which is any of the benchmark concentrations that I just described to you in that last slide. We then compare it to detection frequency and derive bins for our chemicals based off of whether they're likely to cause any kind of ecological risk. So bin 1 here, very unlikely to cause an effect. Bin 6 may be of potential concern within our data set. We then assign bin scores to each of these bins that equate to these bin numbers, and then use those bin scores to prioritize across our individual lines of evidence. So what does this look like in real time when we're evaluating all of our chemicals? Well, each of these dots is a chemical concentration, and each of these individual graphs is the line of evidence under our toxicological potential. You can see for each of them, they have slightly differing characteristics in what exceeds, what doesn't exceed, and how much data is available. And from all of this, we were able to derive bin scores for our chemicals, which we use to compute in further steps. Before we move on to that further step, I have to talk about environmental fate because it is evaluated in the same way. So remember when I was talking about PBT, when we account for persistence, bioaccumulation, and toxicity? Well, this is where it leads back in again. So we just looked at toxicity on a really comprehensive basis, and we also want to consider persistence and bioaccumulation of our potential chemicals. So we take either experimentally derived values or estimates, plot persistence against bioaccumulation, use European standards, which for persistence and bioaccumulation assessment, and then bin our chemicals into similar bins as I described before. So again, bin 6 is this high risk bin, and bin 1 is this low risk bin. So when we take that all together and we plot it in these beautiful heat maps, you can see that we are able to kind of identify which chemicals are rising to the top and which chemicals are rising to the bottom, and which chemicals are associated with significant data gaps. So let's just look at our grab and composite sampling data. All this red up here in this corner here represents pesticide data. So you can see we've got a lot of pesticides that are coming up to the top when we just look at the weight of evidence across our toxicity and environmental fate data. We have on the bottom here kind of a mix of chemicals with significant data limitations. And we've got a mix of kind of lower priority chemicals with a lot of pharmaceuticals and personal care products tending to exist in either this data limited zone or this low priority zone here. Okay, so what happens when we include our other lines of evidence into our evaluation? Looking at monitoring frequency, data availability, as well as these toxicological potential, detection characteristics, and environmental fate. Well, we can identify chemicals that are priorities for effects assessment, so things we need to do more experiments on, chemicals that are priority for environmental monitoring, so things we need to monitor a little bit more, and chemicals which may be priority for management actions based off of the intersection of all three of those. So when we're looking at prioritization for effects assessment, we take the median bin score across all of our lines of evidence and plot it against the available lines of evidence indicating data limitations. This allows us to identify, okay, these chemicals with high amounts of lines of evidence, but low bin scores, which are obviously low priority compounds. These chemicals with high bin scores, but low amounts of evidence, indicating that we should do further testing on them. And these chemicals with high bin scores and high amounts of evidence, indicating that, okay, we know a lot about them and they might still be causing risks. So it's something we got to look at a little bit further. So when we break our actual data down into these different scores, as we've done here, you can tell that it says a lot, right? You can tell a lot from this plot? No, you can't. it's just an open prioritization plot, but I can break it down for you. So basically we identified just in our water, our grab composite data. I'm not going to show you all the data because it gets a little bit confusing. But out of our grab data, out of the 450 detected compounds, we're able to identify about 86 chemicals which are higher priority for effects assessment. And if you kind of push that up to a higher bin score, you can see that the priority chemicals get less and less as well. We're able to identify 173 chemicals, which are kind of in this medium priority score, and 110 chemicals, which are definitely low priority, falling under a medium benchmark score of 2. Finally, we were able to kind of narrow our list down to about 63 chemicals, which have high amounts of line of evidence and relatively high bin scores across all of our available data. When we look at it the same way, but in an environmental monitoring aspect, we can take our medium bin score and plot it against our sampling frequency. So how frequently do we actually look for a chemical within the Great Lakes? And what does that tell us in terms of data availability? So if we break this down again, we can see that we have 239 chemicals, which are in the higher priority category for monitoring, 30 chemicals, which are in the medium priority for monitoring, 110 chemicals, again, low priority across all data sets, and about 72 chemicals, which would be higher priority for management. So this is just under these different lines of evidence looking at sampling frequency and data availability. What happens when we merge that all together? Okay, so we get more data. So we are able to identify across all those lines of evidence what are the highest and lowest priorities across chemicals across the Great Lakes. So if we just look at what we've identified here and you merge, okay, so we have experimental here. This is including our data limitation versus bin score. And monitoring is our sampling frequency versus bin score. Our H2Os are grab samples, our passive is our passive samplers, and we look at the intersection of all of these, and this is 35, but in fact it is 34. We get 34 highest priority compounds across the Great Lakes. This includes 33 pesticides and pesticide degradants, and 1 industrial compound, which is a corrosion inhibitor. We can also do the same to look at the absolute lowest priority compounds across all the Great Lakes, and we find, okay, don't panic, we did have 110, but if we're looking at lowest, that would probably be like, okay, that's definitely low priority. It's 6 compounds. So 2 of these pesticides and 4 of these pharmaceuticals and personal care products. When we also take our data and look at it a different way, identifying which chemicals are high sampling frequency, data sufficient, and have a high ecotoxicological potential, and look at the overlap for that in terms of chemicals that likely would be good considerations for management actions, we're able to narrow that list down to about 22 chemicals from our GRAB data and 17 chemicals from our passive sampler data. Our GRAB data indicated that 4 polycyclic aromatic hydrocarbons are on the list, 6 pesticides, 2 pharmaceuticals, and 10 industrial compounds. And our passive sampler data indicated that mostly 11 pesticides and 6 additional pharmaceuticals are added to the list. So what do we find when we take that all together? Well, just give you a little recap. We're able to identify kind of the highest priority compounds for each of these individual categories. So for research actions and for potential regulatory actions, candidates. So we have a series of pesticide degradants, which honestly are mostly herbicide degradants, which are causing these highest priority compounds across the list, as well as industrial compound, this corrosion inhibitor, 4-methyl-1H-benzotriazole. And when we look at our high priority regulatory compounds, we include some of our classic kind of pesticides, including atrazine, endrin, DEET, some multi-use industrial compounds that have actually popped up in our Milwaukee estuary evaluation, like organophosphate flame retardants and PAHs, and some pharmaceuticals and personal care products like antidepressants. So we have our 2 kind of case studies where I demonstrated in depth how we looked at these chemicals under multiple lines of evidence, looking at multiple lenses in order to figure out which chemicals are priorities across our huge data sets that we're evaluating. So again, we're looking at mixture toxicity, which is complex and sometimes hard to manage. We're looking at single compound toxicity across huge data sets, which we have to really, really integrate and be efficient with when we're trying to figure out how we're going to prioritize. And what we managed to do is kind of break our 80 chemicals in the Milwaukee estuary and 166 mixture groups into 10 single compounds and 7 mixtures that maybe we should look at a little bit further, and 37 compounds and 113 mixtures that we probably don't have to look at as intense in detail. We also took our 550 detected chemicals in the Great Lakes and found about 34 high priority compounds for experimental methods, 37 candidates for further evaluation, and 110 low priority chemicals with, continuous advancements being made in our methodology. And so that'll likely be a little bit smoother once we get our publication out. Okay, so what did we see across all this? Well, there's a lot of data to deal with, right? There's a lot of ways that you can look at things and try and figure out results. And sometimes looking at things, you kind of have to squint your eyes and figure out what everything means and be a little detective. And we also identified, using weight of evidence can kind of help take everything together and help us incorporate both publicly accessible data, freely available software, and kind of all of this complex analysis, and help us identify these high and low priority compounds under diverse experimental settings and across large data sets. So most of this work, we're doing with R, which is free, and we're using all these publicly accessible data sets. So this is something that people could do. You just have to know how to do it and figure it out, which is what we're trying to work at here. So where could we go? Well, we could go anywhere really. So there's a few different things I can think of. We've taken all these benchmarks, we've pulled them together, and pulled together all this code. And once we publish it, it should all be publicly accessible. So that will be like a good advancement for trying to figure out priority compounds across these data sets. We can also take our effects data across the Great Lakes and incorporate it into our chemical data to really refine our chemical assessment, which is currently being done by another group with the Great Lakes Restoration Initiative. And finally, we can take our mixture assessment, refine it a lot, and then use it to evaluate mixture effects across the Great Lakes as well. However, there's many, many, many other options. As you can see, this is an ongoing process, and it's just really trying to take the first steps towards making something complicated a little bit more simple. So I'd like to show you a bit of references, and from that, I'll thank you for listening and take any questions or comments.
Hi, great. I feel like that's kind of a glimpse of the future. We're really entering into this era of science where we're going to have these huge data sets. And I hear so many people saying, well, we have all this data, now what do we do? This is actually one of the first talks I've heard where someone actually has a really logical strategy, intentional strategy to sort that out. So that was really cool. And I feel like a glimpse of the future, right? So thank you so much. So we'll start out with questions from undergraduates.
It was a lot. People need to digest it. They totally can't ask later.
We'll open up to guys.
Yeah, sure.
Can you go a little bit more in depth about how you evaluate the phenobiologic metabolism of some of these chemicals? Going back to your Milwaukee Estuary data.
Yeah, sure. I can pull up the big old xenobiotic metabolism slide and get into it a little bit. Yeah, so that was kind of I don't know if it's like more efficient just to, yep. So yeah, so this is kind of in the hopes of just giving an overview. We looked at expression of metabolic enzymes in liver and intestinal tissue. So we looked at only intestinal tissue and really only in depth in 2017. In 2018, we kind of pared down our analyses. But we looked at expression of like cytochrome P450-1A1, which is typically response to industrial compounds and pHs. We looked at several intestinal enzymes like CYP2A6, CYP2N13, cytochrome P453A, and UDP glucose transferase 1A1 in liver and intestinal tissue. So it's mostly qPCR. And yeah, that's kind of how we evaluated xenobiotic metabolism. In our in vitro effects, we looked at kind of the suite of adagene assays, in which includes these targets for xenobiotic metabolism, kind of proximal, like the aryl hydrocarbon receptor, which is kind of linked to pH exposure as well. And that's kind of how we captured xenobiotic metabolism potential across the, does that make sense?
I think so. Follow-up questions.
Sure.
So how does the adagene assays, like, for example, you have two partners on up there. How does it like determine what genes to look at for certain signal effects?
Yeah, so there's like a standard suite of, you essentially go through the adagene kind of company to get this run. So there's a standard suite of cisfactorial, transfactorial, and GPCR. And the GPCR are more like neuronal pathways there. And these cis and transfactorial just are kind of a suite of like 180 different receptors that they look at. And we just highlighted only the ones with significant responses, which are kind of about an activity like 1.5 fold above the baseline. But actually, there is a way bigger heat map where we just show that basically none of the other pathways have any significant effects, which I can show you if you want.
So each of those lines is actually a group of genes that are
yeah, each of these lines is like a standard protein target, but indicating a signaling pathway. Yeah.
There's one target that's related to one.
Yeah, it's related to like different downstream effects, but yeah. Yep. Yeah. Hi.
Is there any way to look at the spatial district in the Great Lakes study? So is there any effort being put into kind of resolving that spatially, looking at, okay, well, in like the easternmost part of the Great Lakes ecosystem, there's these issues that need to be looked at a little more intensively versus like, well, Superior Neuron,
Yeah, no, definitely. And okay, so the kind of the focus of this prioritization effort was a first pass effort. And then from there, we would identify kind of candidates to look at a little bit more intensively, spatially, et cetera. My role in this, so the research that I presented is part of this big group as part of the Great Lakes Restoration Initiative. And mostly our aim is to kind of take these chemicals and then narrow them down so that we can really investigate them a little bit further. But there's a lot of hydrologists and a lot of people who like are really good at land use assessment. There's a lot of land use data. So that's probably a follow-up step to all of these efforts. Yeah, and I think it's being done by the USGS or something along those lines. Yeah. So yes, we kind of did it for Milwaukee, but I'm not ready to show you those results yet. So yeah.
So if it was up to you,
Yeah.
you had to choose the next sort of, smaller city, I guess, besides Milwaukee, from kind of the bigger database of what area would you focus on?
Well, I'm really looking at the chemical data set on a really broad scale. I'm not really looking at individual samples at all. So that's a tough question. If I was to have an unlimited budget, I would full cannon that and we do a whole Toronto assessment. But yeah, but I don't know what's going on there. So if you guys to find out. Yeah. Yeah, Dan, graduate student Dan.
I was just going to answer the question. Let's see, Toledo.
We'll open it up.
Toledo?
Well, the Maloney River area of concern.
Yeah.
Probably one of the most contaminated here on Great Lakes.
Yeah. That's where, when we was talking about the effects, like folding in the effects assessment and chemical detection, that group, which I'm working as a part of too, we're doing most of our like effects and chemical covariance analyses in the Maloney watershed. So yeah.
So we really are opening up. Anybody have questions?
Yeah.
I'm wondering how these chemicals change just on a seasonal basis, like the presence of some of the chemicals and how that, how they would change. Like if there are some that are not present, for example, in the summer that might be more present in the winter.
Yeah, I can't like speak for the Great Lakes because I haven't looked at that data, but just based off of kind of a knowledge of use patterns, I'd say it's probably you're going to see like an influx of pesticides in the spring after spraying or planting, and then probably you're going to have lower influxes in the winter due to the winter season, depending the geography. And then I don't know, it really depends on the dynamics of the population around it and the industrial operations that are occurring. But that would be my best guess. Yeah, I did my PhD on pesticide toxicity, so for sure in the spring pesticides are coming out.
My question is kind of similar to that, but I didn't get a great sense from your description of the data when these samples were actually taken. And I noticed in the Milwaukee example, the 2 years were really different from each other. So can you just give us a little better, is this like a lot of points in time averaged, or what are we looking at temporally in the data?
Yeah, so Milwaukee, I'm a lot more familiar with the data on like more site by site, sample by sample basis. And in that there was in 2018, it was later in the summer. In 2017, it was earlier in the spring. And across the Great Lakes data set, I think that the majority of the sampling occurred in the spring, summer, and fall. Dan, do you have an idea of?
Yeah, that's correct. And there was different types of sampling. So USGS did what was called surveillance sampling, where they worked across many of the Great Lakes. So usually between 30 or so different tributaries across the Great Lakes each year. And they would rotate sort of different chemical classes. And then there were also more focused efforts by EPA, Fish and Wildlife Service, NOAA, who were focused on certain tributaries, certain rivers, doing more intensive spatial sampling along those tributaries. So there's kind of a mix, sort of a good spatial coverage, but then there's more intensive sampling in certain areas. So it's a really heterogeneous data set in terms of how intensely certain areas were sampled. And that's part of where, again, getting into that more spatially explicit analysis of the data is going to be important in these initial prioritizations.
Yeah, thank you, Dan. In case you did know, Dan is leading the project. So. But I appreciate that. Yeah, so yeah, it's really an amalgamation from a multi-agency research effort. And my part of the process has really been just this kind of like large scale weighing the chemicals down and less focused on the distribution, and kind of spatial, less focused on the spatial stuff, which will be the next step in the analysis for sure.
Okay, I actually think we'll have to move any other questions to after the seminar, so please come on up. And put together, thank you again for the